This presentation is about frameworks and tools for soft tissue morphometrics in the human knee. I'll start with some background information and explain why we need soft tissue morphometrics. Then, recent related works will be summarized. I'll introduce our approach for knee cartilage and meniscus morphometrics. These images show the development of cartilage lesions with the progression of knee osteoarthritis. Studies have shown that cartilage degeneration happens at the early stage of knee osteoarthritis. These images show another type of knee disorder, meniscus lesions. Figures on the right show 4 kinds of meniscus lesions. The appearance of meniscus lesions is another indication of early knee disorders. You might wonder why we need soft tissue morphometrics. Aren't imaging techniques like MRI sufficient for soft tissue analysis? The answer is probably no. Since morphometrics can provide quantitative measurements, it is desirable to derive these metrics from medical images. We summarize the benefits of soft tissue morphometrics from the perspective of patients and clinical practitioners. From the patient's perspective, it allows for early detection of knee disorders, which enables timely treatment and progression prevention. Quantitative information on soft tissue abnormalities enables personalized surgical planning and treatment. On the other hand, it might alleviate the burden on clinical practitioners through automatic image analysis that provides more informative imaging features. This enhances the quality of healthcare services by enabling risk stratification to identify individuals at risk. Other benefits include disease monitoring, clinical studies, the development of new imaging techniques, data analysis methods, treatments, and drug discovery. Most deep learning researches in this field focus on segmentation. This is reasonable since segmentation highlights the region of interest and provides anatomical information. It is considered the first step of most medical image analysis tasks. Here we summarize recent advances in cartilage and bone segmentation from knee images. The prevalent model type is convolutional neural networks, with an increasing number of transformer-based models and semi-supervised or self-supervised methods. Fortunately, in recent years, we have seen the emergence of models with excellent generalizability that perform well on data from unseen domains, such as the NNU-net and MEDSIM. With these auto-configuring or foundation models, segmentation has become a relatively straightforward task. While we are impressed by the performance of segmentation models, it might be wise to pause and ask a few questions, such as whether segmentation is all we need. Starting from the question of what defines a good segmentation model, it's important to notice the pitfalls in metric selection. Different metrics capture various aspects of model performances, and downstream tasks may demand varying levels of model accuracy. For example, a volume measurement requires a volume-based metric, while a boundary-based metric is more suitable for surface measurements. With competitive foundation models available, there may be a desire to redirect our focus towards research fields that facilitate clinical applications, such as quantification tasks, automatic data processing, software design, and other deep learning research. One example of interesting deep learning research is counterfactual image synthesis, where the what-if type of question is answered. For instance, we might be interested in what the knee MR image would look like assuming 50% increased lesion in the future given the current scan. Here, I summarize relevant studies in deep learning and medical image analysis for knee osteoarthritis. Deep learning-based image classification tasks mainly include severity grading and phenotype classification. Some registration models have been developed on knee image datasets, and there are semi-quantitative or semi-automated quantification methods. We have identified a research gap in the development of practical applications leveraging advanced deep learning models. Creating accessible and user-friendly tools from these models is essential. Can we bridge the gap between images from the scanner and the quantitative metrics required in clinical practice? I'll first introduce our approach to cartilage morphometrics. We introduced a framework called CartiMorph, designed to generate regional quantification metrics from knee MRI. This framework comprises 3 models, responsible for image segmentation, registration, and template learning. We developed algorithms for thickness mapping, lesion quantification, and subregion partitioning. These components are seamlessly integrated into an automatic image processing pipeline. We have made the code available on GitHub. The figures in this slide show the network architecture of the template learning model, the learned template images, and the proposed lesion quantification algorithm. For further methodological details, experimental setups, and results, please check our paper. We have developed a standalone software called CartiMorph Toolbox. It implements the CartiMorph framework and adopts a few improvements including auto-configuration of the computing environment, image standardization, and improved model performance. Models training, fine-tuning, evaluation, inference, and sharing are supported. Additionally, it has a built-in data visualization module. We have released the toolbox and made the trained models available on GitHub. We also built a standalone version of the visualization module called CartiMorph Viewer. It may help with data sharing across collaborators. With the viewer, users can check the intermediate results and the final metrics. The viewer is also available on GitHub. In this section, I will briefly introduce some useful tools for the development of a meniscus morphometrics pipeline. The NNU-net and MEDSIM models provide a strong foundation for segmentation model development. Additionally, an interface for NNU-net has been developed. Existing mesh processing libraries can be used for downstream tasks. Finally, we present some exploratory approaches for meniscus morphometrics, including the quantification of coverage, extrusion, and thickness mapping. The two figures on the right display our preliminary results. I am currently supported by the CDT in Biomedical AI at the University of Edinburgh. Part of the work introduced was done when I was with the Chinese University of Hong Kong. Feel free to reach out to me for further discussion.